Professor Wan-Seob Cho from the Laboratory of Toxicology, Department of Medicinal, Biotechnology, School of Natural Resources and Life Sciences, Dong-A University, Busan.
Thank you, Chair, for your nice introduction. And I want to thank the organizing committee for inviting me for this nice meeting. So today, I want to talk about the impact of size and surface charge on the kinetics of gold nanoparticles. So as you all know, there are many physicochemical properties can influence into the kinetics of nanoparticles. For example, shape and crystallinity is important, and size or length is important. So smaller size can distribute more wide than a bigger one, or the long fibers act differently than tangled or short one. And compositional element is also very important. For example, gold nanoparticles have accumulating properties, and silicon nanoparticles usually excreted fast. And solubility and biopersistence is important because high solubility nanoparticles can easily cleared more faster than low solubility nanoparticles. And surface charge and surface reactivity is important. So there are a huge number of factors can influence the kinetics of nanoparticles, but there is not much studies about to evaluate about those factors. So in this study, we evaluate the law of composition and the law of size and law of surface charge of nanoparticles on the kinetics of nanoparticles. And those information can be used for modulation of physicochemical properties to improve the kinetics of nanoparticles. So for evaluation of size effect, we synthesized pegylated gold nanoparticles having 30 different sizes for 13 and 100 nanometer in size. And those nanoparticles were injected into the intravenous to the biopsy mouse at 0.85 milligram per mil body weight. And after each time point, several samples were collected to evaluate the concentration of gold by ICP mass analysis. So until 24 hours, 4 and 13 nanometer gold nanoparticles shows higher concentration than 100 nanometer gold nanoparticles, and especially 100 nanometer particles shows almost cleared even at 30 minutes after injection. But after that, from the 7 days, all particles was cleared, almost cleared. And in the tissues, when we injected 4 nanometer gold nanoparticles, the main organs or accumulating organs was liver, spleen, and lymph nodes. And interestingly, those organs, the concentration of gold was continuously increased up to 7 days after injection. After that, it slowly cleared, but even at 6 months after injections, it still have high concentration. And other organs such as lung, kidney, heart, and testis, and brain shows low levels and some clearing pace. For 13 nanometer gold nanoparticles, it shows similar pattern with the 4 nanometer gold nanoparticles, and the concentration was peaked up to 7 days after injection, and it slowly cleared, but it also have a high concentration even at 6 months after a single injection, and other organs has low concentration. And for 100 nanometer particles, the concentration was a little bit flat, and the concentration at the 30 minutes after injection, it is very high compared to other time points. And it also shows accumulating properties. And other organs has low concentration. And especially 100 nanometer particles shows lower concentrations than 4 or 14 nanometer particles. So bigger particles shows less wide distribution. So, and to evaluate the subcellular localizations, we did TEM analysis at 7 days after injection, and we found in the liver, we found the gold nanoparticles in the Cooper cells, especially inside of the phagolysosomes, and all those particles were aggregated or agglomerated inside of the lysosomes. And in the spleen, it also shows particles was found in the macrophage-like cells, and lymph nodes also we can find the particles inside of cells, especially macrophage-like cells. And excretion was evaluated at each time point, and this is urine and this is bile. And we found that the concentration was really low, but it is very consistent with the accumulating properties of nanoparticles. So this study was to evaluate the surface charge, impact of surface charge on the kinetics of nanoparticles. So we synthesized 15 nanometer gold nanoparticles having basically PG. And from the PG gold nanoparticles, we conjugated the carboxyl group or amine groups to have negative charged or positive charged particles. And all has similar sizes. And with those particles, we injected into the tail vein of barbacy mouse and evaluated gold concentration in each organs at each time point. So all particles shows similar size distribution, and all particle has a PG as a basic, and but they have a different surface charge. So when we summed total gold nanoparticles in all measured organs and evaluated the percent of total gold compared to initial dose, we found that until 24 hours, it shows differential between the surface charge. So in case of pegylated one shows less concentration than others. But however, after 7 days, it shows similar concentration regardless of the charges. But even at six months, about 30% of golds were still remained in organs, not excreted. So in the blood, we found that pegylated one has a longer blood circulation time. But even aminated one or carboxylated one, they all have PG, but they are cleared very fast than only PG pegylated gold nanoparticles. So pegylated particles shows higher circulation than others. So in the tissues, we found in the liver, carboxyl group has a higher concentration than amine or pegylated one. And in the spleen, the aminated one was higher than others. And in the lymph node and the kidneys, we found pegylated one was a higher concentration than others. And in the lung and the heart, we found amine group. Amine group was higher than others. And in the brain and testis, the pegylated one was higher than others. So for excretion pattern in the urine, even the concentration was relatively low. The major route was different. In case of PG1 and amine1 was the main route was the main excretion route was urine. However, in the bile, carboxyl group was mainly excreted by in the hepatobiliary clearance system. So, as a summary, the smaller nanoparticles, gold nanoparticle shows longer blood circulation time, and all gold nanoparticle shows accumulating propensity more than 6 months. And likewise, gold nanoparticle shows minimal excretion in both urine and bile, and most of nanoparticles were localized in the reticular endocellular systems, such as liver, spleen, and lymph nodes. And for impact of surface charge, pegylated one, just for example, the neutral one showed longer blood circulation time than others. And the organ distribution of gold nanoparticles shows functional group specificity. And all gold nanoparticles showed low elimination rate, but showed differences in the route of elimination. Thank you very much for listening.
Thank you very much, Professor Cho. The lecture is open for questions, please.
I have a couple of questions. Was the injected dose, could it be that the injected dose was enough to saturate some of the uptake systems?
Sorry, can you say that again? Injected goals was?
Could it be that the injected dose was sufficient to saturate the uptake systems?
Saturation, you mean? I think the dose 1 milligram per kilogram body weight is very low. So the injection was just a single injection. So I think it cannot be saturated in organs.
Okay, and the next question would be, you showed that there was uptake in Cooper cells.
Yeah.
What was the cell marker? How did you come to that conclusion that the uptake was in Cooper cells and not other types of liver cells?
You mean, how can I distinguish the Cooper cells?
How did you distinguish the Cooper cells?
Yeah, actually, that is done. Actually that was discussed with the specialist of special pathologist. So I advised from them. So yeah, pathology.
I would expect a lot of the uptake to be in the endothelial cells, but that's another story. We can't discuss that now. My last question would be, I would expect also that a lot of the uptake would happen before a half hour after injection. So it would be interesting to see the uptake kinetics before 30 minutes.
That's a good point. Because in case of 100 nanometer particles was cleared very faster within 30 minutes. But it is very difficult to make sacrifice animals within 30 minutes because yeah, that's a good point, but it must be very difficult to make an exact time point.
But I would actually expect that most of the uptake was done when you measured it at 30 minutes.
Yeah, in case of when the amine group or carboxyl group gold particles was almost cleared within 30 minutes, but in case of pegylated one was have a longer.
Thank you for your talk. When you compare the clearance in the liver and in the spleen, so you had 3 different sizes.
Yeah.
Now, if you take the 4 nanometer and compare it to 100, your conclusion was the 100 was cleared much faster than the 4 nanometer.
Yeah.
So it was prolonged circulatory half-life for the 4 nanometer. When you double the radius, you're increasing the volume of the particle by a cube to the power of 3. So comparing a 4 nanometer to 100 nanometer, that means for every 100 nanometer particle, you have 100 times more of the 4 nanometer particle. So your dosing is based on the weight, but not on the number of the particle. So you have 100 times more of the 4 nanometer particle. So what happens if you do the correction?
Yeah, that's a good point. I think because we injected particles based on the mass dose, and we found the gold particle, bigger one was cleared more faster, and that was the concentration of gold, not of gold nanoparticles. So, so when we converted to number of particles, it must be more exaggerated in case the 4 and 1 smaller one has a more number, even it has a lower concentration. So it must be more exaggerated, more, yeah, I think. Half-life in the bloodstream. But 4 nanometer shows consistent level up to 24 hours.
Because you have 100 times more particles.
But that is, so I injected 4 nanometer gold nanoparticles at the same dose and sacrificed the animals at the same, each time point. And compared, so the injection dose or injection size is the same in all.
The weight of the particles would be interesting if you give the same number of particles between the 2 cycles. Then you will see the real difference on the pharmacokinetics.
Yeah, thank you.